O-Level Biology 5090, Chapter 18, Biotechnology and Genetic Modification, Part 1. And in this part, we will discuss biotechnology. Before going in detail, I would like to discuss the equation shown on the screen. And in this, the process of fermentation is shown there. In this equation, the process of fermentation is taking place. And what is fermentation? Fermentation is the anaerobic respiration which takes place in the absence of oxygen. So in this equation, you can see that yeast cell is converting the sugar molecule into ethanol and carbon dioxide. And as a result of this conversion, small amount of energy is produced in the form of ATP. So I would like to say that during the process of fermentation, the yeast cell will convert the sugar into ethanol and carbon dioxide by release of small amount of energy. And in the next slide, and in the next slide, we will discuss the role of yeast in the production of bread and ethanol. As students, you know, in the previous slide, uh, the yeast cell was converting the sugar molecules into ethanol and carbon dioxide. So here you can see that how can we make use of this process of fermentation at home or at commercial level. So for this purpose, uh, flour, yeast, and small amount of sugar, they are mixed together by using water, and it will help to make a dough of this mixture. So dough will be kept for some time. This dough will be kept for some time for the process of fermentation, and after some time what will happen, the dough will change. As you can see in the picture, it will raise and there will be small bubbles inside. So what is happening in this process? That is, the carbon dioxide gets trapped within the elastic network of gluten strands in the dough. So carbon dioxide will produce these small bubbles into them. As the gas builds up, it causes the dough to expand. So due to the formation of these bubbles, it will help this dough to expand. This process is known as proofing. So proofing is what? When the bubbles are produced in the dough and it makes the dough to rise. The ethanol produced by the yeast contributes to the flavor of the bread, or this ethanol which is produced as a result of fermentation, it is evaporated. It is evaporated. Only carbon dioxide will produce the bubbles and ethanol will be evaporated into the air. So in this way, the yeast help in the production of bread and ethanol as a result of fermentation. Next question, or you can say bacteria are useful in biotechnology and genetic modification. So we can not deny the role which the bacteria are playing in both of these fields, that is biotechnology and genetic modification. So bacteria are particularly good at accepting foreign DNA. So they are very good in accepting foreign DNA, and introducing genes into the bacterial cells is now routine. The process of, don't forget the process of genetic engineering, by which we can making use of the you can say making use of these bacteria in deleting, in altering, or in inserting the genes into the organisms. And this procedure is a first step to making bacteria that can do new and useful things. So bacteria that contain introduced DNA are classified as new genetically modified organisms. So The bacteria which we use for this purpose, when the foreign DNA are introduced into the bacteria, in that case, they are called as genetically modified bacteria. So these genetically modified bacteria are very helpful. Genetically modified used, so they are used to produce large amounts of proteins for industrial use. So they can be used for different purposes. One of them is the production of this protein for industrial use. Generally, the bacteria are grown into a large volume before the gene encoding the protein is activated. So the bacteria are then harvested and the desired protein purified from them. So with the help of some processes, we can make use of these bacteria in the different fields of life to get other products. So because they are, it is very easy for the bacteria to get inside the foreign DNAs. By using this foreign DNA, the bacteria are genetically modified, and genetically modified are bacteria are very helpful for us in different fields. So examples of these use of these bacteria are microbial biotechnology applications include the use of bacteria to produce antibiotics. We are making antibiotics by using of these bacteria and other pharmaceuticals, the use of yeast to produce food and beverages, and the use of fungi to decompose organic matters in landfills. So in this way, not only the bacteria, the other microorganisms, as for example, yeast and fungi, are also helpful for us in the production of different products. The next, why bacteria are useful in biotechnology, genetic modification. We are making use of these bacteria in different fields, as we are simply we are discussing at this time the biotechnology and genetic modification. So the bacteria are useful in both of these fields, that is in biotechnology and genetic modification, because they can be grown and manipulated without raising ethical concerns. They are bacterial cells. There will be no issues with the ethics. So they can be manipulated, they can be grown without raising any ethical concerns. They have a genetic code that is the same. Number one, and number two is the genetic code, which is the same as other organisms. So scientists can transfer genes from other organisms or plants into bacterial DNA. So it is very easy to for the scientists to transfer the genes from other organisms. As for example, the other organisms are animals or plants, and they can be transferred these genes to the bacterial cells. And why it happens, that is the bacteria multiply after every 30 minutes. So they are doubled after every 30 minutes. So in this way, their multiplication rate is very high, due to which the product can be, you can say, get in or can be harvested after a very short time. They so fast 2 to 3 times an hour, bacterial DNA is in the form of a circular strand, circular strands. So circular strands, the bacterial, don't forget this bacterial. You can see chromosomes, which are without nucleus. They are embedded into the cytoplasm in the center. And beside this chromosome, there are other circular DNA, which are called as plasmids. So these are plasmids. So that is the reason they are useful in both of these fields, biotechnology and genetic modifications. Now, when we are producing the products on large scale, we need large pots for it. So large pots for the process of fermentation, we can have large containers which are, you can say, rustless. They are made up of stainless steel. And for example, the product which we can manufacture into them is the antibiotic. Penicillium mold for producing penicillin. So penicillin is an antibiotic. We can produce insulin as well. So many other products can be manufactured on large scale in the fermenters. Now in the next slide, you can see a large fermenter. We can discuss its different parts. So first of all, the place from where the nutrients and microorganisms are added into the fermenter. So this is the in this green arrow shows from which the nutrients and microorganisms are added into it. And then you can say the water cooled jacket is outside. So this water cooled jacket is there to maintain the water temperature, heating, cooling water out. So water, if we are taking out the water, we want to take out the water. So this point will be used in this purpose. So the other, you can say the lower part, it will show when we want to enter the water, we want to enter the water into the fermenter. And on the other side, we can have sterile air supply. Sterile air supply means air without oxygen. Air without oxygen. So this type of air is called as sterile. And then stir it. Stir it to keep the microorganisms, nutrients, and temperature evenly distributed. Means it is used to mix the whole nutrients or which are used at this time for the process of fermentation. Now, the whole process can be monitored with the help of a computer. And with the help of computer, we can monitor what is taking place inside the fermenter. Beside all this, the temperature and pH should also be the monitored. It should also be the maintained because the different products are manufactured at different pH level at different temperature. So the pH and temperature play a very important role. They should be maintained for the product which is being manufactured in that fermented at that time. So the product is harvested, which is the point which is at the lowest of this fermenter. So in this way, the fermenter is used for the process of fermentation. So the different factors which should be keep in mind before fermentation, that is, the fermentation of different products can take place. With the help of this fermentation, we can manufacture different products. First of all, antibiotics, formation of insulin. Thirdly, we can manufacture the different edible products as, for example, cheese, yogurt. So in this way, the different pH and different temperature is required for all these products. Most of the organisms used in the fermentation are aerobic. Sometimes in the process we need oxygen because aerobic metabolism is more efficient than anaerobic metabolism. So both of these, aerobic and anaerobic, both of these processes can be, you can say, continued into this fermenters. So oxygen demand, whether it is demanding, we are going to produce a product. We must know whether oxygen is required for that process or not. And then consistent pH. Consistent pH, if the pH we need is 6, so it should be 6. It should not exceed from 6 or it should not decrease from 6. So it should be maintained. Same with the temperature control. Temperature, if we need 26-degree temperature for the process, so there should be the temperature 26 degrees centigrade. Rate and nature of mixing. So whether we have to use the stirrer to mix the ingredients which are present in the fermenter or not. So it depends. A supply of nutrients for rapid growth. Nutrients should be supplied proper quantity at the proper time. Anti-foaming agent to alleviate excess foaming. Anti-foaming agents should be added because foaming is produced it can alter the metabolic rate inside the fermenter, so antifoaming agents should be used. Moreover, optimal conditions generally change with each tenfold increase in the volume of a bioreactor. Simply, we can say the different environmental pH, temperature are required for the formation or the manufacture of different products inside the fermenter. Now, the next we will discuss the use of pectinase. Use of pectinase for food juice production. Pectinase is an enzyme. So, pectinase is an enzyme which is used in the fruit juice production. So fruit juices are extracted using an enzyme called pectinase and pectin, because pectin is a substance which helps to stick plant cells together. So plant cells, they are joined together with the help of this pectin. They are, you can say, sticked together. A fruit such as apple or orange contains a lot of pectin. So pectinase breaks down pectin. So the pectinase will break down the pectin, and then this makes it much easier to squeeze juice from the fruit. So when pectin will be broken down with the help of pectinase, so it will be very easy for us to squeeze the juice from that fruit. So pectinase also helps to make the juice clear rather than cloudy. So juice which is made, it should be cloudy. It is cloudy, but pectinase will help to make it clear. So the cloudiness in fruit is often caused by insoluble carbohydrates, which load in the juice. So the pectinase and other enzymes such as cellulase can be added to the cloudy juice. So pectinase and cellulase can be added. So they will break down the insoluble substance to soluble one. So the juice becomes clear. So we can have the clear juice by using this pectinase enzyme, which converts the pectins or break down the pectins. So in this way, we can get a clear juice from the fruit juice products, apple or other fruits. So here on screen, you can see two types of juices. One of them is juicy, other one is clear. So the apple juice in the left hand bottle has been treated with pectinase. So it is treated with pectinase. So it is clear. The other, it shows the cloudy. It means it is not um, treated with pectinase. Now, the use of enzymes in biological washing powders. We use washing powders to wash our clothes. So washing powders contain detergents. They help insoluble fats, oils mixed with water. When we are at home or outside, sometimes we have different stains on our clothes. So these stains can be of fats or oils, proteins or blood stains, etc. So different types of stains, juice, tea. So we can have different types of stains on our clothes. So these stains are removed by using different enzymes in biological washing powders. So biological washing powders contain these enzymes as well as detergents. So these enzymes help to break down other kind of substance which can stain cloths. They are especially good at removing dirt which contains colored substance from animals or plants like blood or egg stains. So these different types of stains are removed by using these enzymes. So the enzymes, they are of different types. Some enzymes work best for carbohydrates, so carbohydrates, and the other proteases. lipases. So all they are added into the biological washing powders to remove the stains from the cloths. And in the next slide, we will discuss about them. How do they work? So some of the enzymes are proteases. Proteases means the which break down the proteins into amino acids or into insoluble soluble components, which catalyze the breakdown molecules into amino acids. This helps with the removal of stain caused by proteins such as blood stains. Hemoglobin. Hemoglobin is a protein, so blood stains cannot be removed from the cloths until we make use of these enzymes into them. So enzymes are there. This protease is biological washing powders break the hemoglobin molecules into smaller molecules, which are not colored and which dissolve easily in water and can be washed away. So it is very easy for us to remove these stains. Some of the enzymes are lipases, which catalyze the breakdown of fats to fatty acids and glycerol. This is good for removing greasing stains molecules. So in this way, I would like to say that as enzymes play a role in our lives, when in the you can say in the process of digestion, now they also play a role when we have stain on our clothes. So these enzymes will break them down into their basic components and which will be washed away by using water. Another use lactase for lactose-free milk. Some people are there just like me who cannot digest the lactose. So for those people, there can be the lactose-free milk. So lactase in enzymes produced by many organisms. So lactase is enzyme which helps to digest the lactose. So it is located in the brush border of the small intestine. Brush border where the finger-like projections are there. Of human and other mammals. Villi, they have inside the villi, they are located inside the villi. It breaks lactose down into two small sugars, glucose and galactose. Glucose monosaccharides, both of they are the monosaccharides. So these monosaccharides, they give sweetness to the milk. So it is essential to the complete digestion of whole milk. So the milk uh, lactose in the milk, lactose will be broken down into sugar, simple sugar, which is glucose and galactose. So lactose-free milk is made with regular cow's milk. Where it differs is that it has the enzyme lactase added, which breaks down the lactose in milk. So only lactase is added into that milk, which will help to make a lactose-free milk, making it both easier to digest and helping in avoiding the symptoms associated with lactose intolerance, such as stomach pain and nausea. The people who are intolerant to the lactose, so they will have the pain and nausea and stomach. So all these symptoms can be removed by using lactose-free milk. So students, uh, it was all about biotechnology according to our syllabus. So please like, share and subscribe my channel, and thanks for watching.